I want to thank a special guest for this upcoming podcast. Dr. Sarah Onida is a consultant vascular surgeon and honorary senior lecturer in vascular surgery at the Imperial College in London. Dr. Onida has been prolific with over 100 publications over the last 13 years, and she's a worldwide lecturer. She's fluent in 5 languages and truly one of the rising stars in venous disease. Thank you for agreeing for this podcast.
Thank you for having me. I'm delighted to be here today.
So, could you let us know what is metabolic profiling for venous disease? You've been working in this field for a while.
So that's absolutely right. It's sort of my field of niche interest and really stemmed after my PhD, where I worked a lot with metabolic profiling platforms. But basically, it's cutting edge technology that isn't probably as novel as it was maybe a few years ago. But as opposed to looking at the genome or the proteins, when looking at disease pathophysiology, it looks at end products of cellular metabolism. So you've got specific experimental platforms that can detect very small sugars, very small amino acids, very small lipids. And those are really the end products of what has happened in a cell, as opposed to what the cell is coded to do, which is what many of the platforms that we do have and have been using for the last decades really tell us. So it's very interesting because it can really give us a good idea of what biological processes have happened in disease, and it's much more representative of the gene environment interactions, and clearly in conditions where the gene environment interactions are important, such as in venous disease, whether that be thrombotic or whether that be superficial venous reflux. This is clearly quite useful technology to help uncover some other areas that are still unclear of the pathophysiology of a specific condition. So I find it really exciting because A, it's a new way of looking at things, but B, the technology in this field is constantly changing and we're getting much better at point of care testing, which I think is really exciting for patients.
So, how do you see this field helping us diagnose and manage venous disease?
So I think it's important to mention that actually much of the research that has been performed using these platforms has been in cancer research. That's where the greatest investment has been. And that has been from response to chemotherapy and large-scale oncological studies to actually looking at surgical resection margins when performing surgery to remove cancers to make sure we detect where the safe margin is. And really that is sort of working in conjunction with a surgeon in theatre to get immediate results so that you don't have to wait for the histology to come back before you find out whether your operation has been successful or not. So this is really interesting and has been developed quite a lot in cancer research, but has been used a lot less in vascular and venous disease, although there are units both in Japan and in China and in the UK clearly that have been championing this technology specifically. With respect to venous disease, I think it's always very interesting that we're dealing with something that was mentioned back in Egyptian times, and yet we're still fighting with this disease, trying to optimize its management. And although we have developed novel technologies to address superficial venous disease, for example, in terms of endovenous ablation, in terms of the latest technologies that have come out, the basic tenets of removing the refluxing trunk haven't really changed compared to the old-fashioned high-tie and stripping. And although we are much better at managing things, we can't stop the disease from occurring, we can't stop it progressing, and we can't stop it from recurring, which are the main areas that are of interest. And I think metabolic profiling might be quite helpful in these, specifically because it's very good at personalizing or characterizing a specific individual's phenotype. So over time, with using it more and getting more data points, we might be able to develop models to then be able to predict people who are more likely to develop the disease, or are more likely to develop patterns of recurrence, or are more likely to have complications or develop ulceration. So I think that's one area where it's really useful. I think it's incredibly useful in terms of biomarker development, particularly in the deep venous thrombosis area of research. We know that D-dimer isn't great. We're looking at other biomarkers at the moment, and actually this is really good technology to try and test some of the more novel and nuanced biomarkers. And finally, with respect to wound research as well, patients with venous leg ulceration have such a hard time healing, and identifying phenotypes of healing versus non-healing ulcers, regardless of what their hemodynamic status is, regardless of whether they've had all their superficial venous reflux treated, is really important in terms of targeting therapy and treatment to the patients who need it the most. So I think there's lots of remits where it can be used, and actually it's very exciting technology.
Well, can you keep using the word technology? Can you just describe some of the new technology that's coming on board that's helping look at this particular field in venous disease?
So classically, when you describe metabolic profiling, there are two major types of experiments that you perform. There's nuclear magnetic resonance spectroscopy and mass spectrometry. And basically, they use slightly different experimental modalities to look at the same thing. They're like two sides of one coin. One of them is better at looking at amino acids, and the other one's better at looking at lipids, for example. So together, by using both experiments, whether you take tissue samples, whether you take plasma, whether you take urine, those machines are able to assess these, and they give you a global metabolic profile. So they basically tell you what metabolites, what little biomarkers are present in those tissues or in those fluids that you're getting. Now, the difficulty with those is that although they're very precise, the experimental time is quite laborious. So an experiment takes a number of days. If you have a lot of samples, you can't run the experiment until all your samples are collected, and the preparation time is quite long, and the samples in some cases get destroyed when you analyze them, so you can't use them again. So although it's very helpful technology, it's not the most user-friendly, and certainly isn't an easy thing to use if you want a point of care test. But over the years, the ambient mass spectrometry has been developed, which is a field of research where actually, I won't delve too much into the chemistry of mass spectrometry, but it's based on ionization. So you ionize a sample so that you can then detect its chemical composition by breaking it down. And in ambient mass spectrometry, you're able to do that very quickly and very easily, just as a point of care test, without having to go through all the experimental motions that you would with classical mass spectrometry. So it's a much easier, user-friendly technology to use. And now that that is available, we have been using it in theatre during ulcer debridements, in clinic, coupled with a laser, to try and look at what the biomarker signature is in a specific patient as a point of care test. So it's much more accessible now. So I think the newer technology is certainly going to be a lot easier to use for patient care than the standard technology that just looked at the biomarkers.
I really like the idea that you're extending what we are already doing with looking at technology that can look at a biologic, at a microscopic level. I think this really it extends what we know and what we do to a whole new field. So hopefully it'll be something interesting. But how could this be cost effective with respect to, let's say, global venous care?
That's a really important question, and this isn't cheap technology, clearly, and it's not technology that is easily accessible everywhere at the moment because it is limited to large sort of universities and research units. But I have to say that over the last 10 years, the cost of running an assay has reduced as more assays have developed. So it's a lot less expensive than what it was initially. But the way I see this being helpful is in venous disease is twofold. From one perspective, I think it's a really powerful way to look at the pathophysiology in detail. And therefore, by using patient samples and collecting patient samples of patients with different seep stages and coupling it with other platforms like protein work, like genomic work, I think we can really characterize what biological pathways are occurring in different presentations of venous disease. And I think that's very important to help our understanding of the disease process, because although there is, we know loads already, there are many other nuances that we're still working on. And I think this might be a good way of trying to develop and understand those better. So the pathophysiology aspect is one area where I think this is going to be helpful, and that will cost, the cost of the assays, I guess. But in terms of bringing it to patient care, which I think is the most important part, because much of the basic science work that has been performed hasn't really translated into patient care at all, really. What this technology allows us to do is, by developing specific ways of taking fluid samples from an ulcer, for example, or assessing the skin of the patient using non-adherent dressings, for example. We can develop ways of using these media that can then be collected and sent off to be assessed very quickly. And ultimately, I guess what you could develop is, a point of care test where it's almost like a litmus test, where the little swab that you use will turn a specific color if a specific metabolite is detected. And say if you had a patient with an ulcer that is present and you want to know whether this is going to be a hard to heal ulcer or it's going to be an easy to heal ulcer, you could just use a swab that has been formulated specifically to detect certain metabolites, pop it on the ulcer, and it could change a color, and potentially that can inform the way you manage that ulcer further. So I think the benefit of this from a cost effectiveness perspective will be downstream to some of these media that we will develop and are currently working on to try and detect those specific metabolites that are either harmful or a poor prognostic sign, for example.
You're clearly one of the leading experts in this field. Can you tell us about your journey in this field with respect to your research?
So I started off with my PhD. So I was a vascular trainee and got into the department at Imperial under the mentorship of Professor Davies. And there was previously some work being performed with respect to metabolic profiling, and we've been very lucky to have collaborated over the years with people like Jose Diaz and Joe Raffetto as well with these mechanistic studies. And then I just took it on when it came to varicose veins and superficial venous disease. And then over the years, as I then went into an integrated clinical academic training scheme, throughout my training, I was then able to continue working on this. And now I'm supervising 5 PhD students while working on different aspects of this and developing this further. And a few of these are in venous disease, and you may have had the presentations at some of our conferences. So no, I've been lucky that I've been in a supportive environment and have had access to this technology at Imperial. And so it's a very exciting place to be in.
So for young researchers or people who are just starting their early career, what would you suggest for starting their career in translational research or basic science? Or you're clearly a leader in this. What words of wisdom would you give a young person who's starting out?
I think when I meet prospective PhD students who may or may not want to do translational research, in our department, we do a lot of clinical research as well, and I'm heavily involved with clinical trials as well. And students sometimes come and say that they're very worried about doing a basic science project or very worried about doing translational research because they may not have been in the lab before, or they might feel that this is a little bit more daunting or less, they're less used to it than perhaps clinical research or recruiting patients for clinical trials. I would just say not to be daunted. It's a fantastic opportunity to develop something that is quite unique. You get the chance to work with some of the most interesting and intelligent people I have ever met, and many of these are analytical chemists. And being able to bring your clinical knowledge into the lab and have their lab knowledge and give them a way of applying it to patients is absolutely fantastic. And it's just really interesting and stimulating working with people from lots of different backgrounds. So I would just say don't be daunted. If it's something that you might be interested in, absolutely go for it. And it is very rewarding, but also very, very hard work. And clearly experiments sometimes go well and sometimes they do not. And it's important to not take no for an answer and not give up and keep on going. So I'd encourage anyone to do it. I think it's really great.
Thank you so much for doing this podcast. It's been quite enlightening, and I'm really looking forward to the future research from your lab.
Thank you very much.